Welcome back to Environmental Epidemiology, and in this video we're going to speak to the SARS-CoV-2 testing protocol that's used for determining a confirmed case of COVID-19. And before I go into too much detail about that, I just want to start off, as I do occasionally with classes, with a quote from Dr. King. And Dr. King said, "In a real sense, all life is interrelated. All men are caught in an inescapable network of mutuality, tied in a single garment of destiny, and whatever affects one of us directly affects all of us indirectly. I can never be what I ought to be until you are able to be what you ought to be, and you can never be what you ought to be until I am what I ought to be." So we all are in this thing together. Not just with this class, but as a country, as the Commonwealth. And this, as Dr. King said, is the interrelated structure of reality. So we're all in this together, and in this set of slides here, I'm going to show you this image again that may be something that's hard to look at, but a reality for folks who are going to be going through the SARS coronavirus testing. So let's take a look at this image. What do you see there? That is a nasopharyngeal swab. That nasopharyngeal swab that you see there is going all the way to the back of the nasal cavity where it meets the pharynx. And this is the most sensitive method that we've got for actually detecting the presence or absence of the actual SARS coronavirus 2. There are other places in the body where they have been able to actually identify the virus. Mucus, sputum from the deep lung, even saliva, and also even feces. So the stool samples have actually had some SARS coronavirus in some cases, at least based off of what we know from the Chinese data. However, that region, that area here in the back of the nose where it meets the pharynx, that area is the most sensitive, meaning that if a person's carrying the virus, that area is the most sensitive in the sense that if they truly have the virus, we're most likely to actually find it by looking there. So we don't want to waste the test on a saliva sample or a mucus sample and get a negative and then give a person a false sense of security that they're okay, and then they go about and infect others. Or they're not monitoring their condition enough, and they may end up in a situation where they should have been monitoring it and may have actually needed hospitalization. So getting into how that test is actually used. So what happens to that swab after we're done with it? Does the swab get dipped into a liquid solution and change a color? Because many Americans very well may believe something like that, because we grew up watching CSI and watching these other shows that have made science, although it may make it look more appealing and attractive to many people, they also make it look simpler than what it truly is. So this is not what we actually do. The actual WHO protocol is for a 96 well plate or a 256 well plate or a 512 plate, but overall the WHO method is for us to do reverse transcriptase quantitative polymerase chain reaction, or RT-qPCR, which is also sometimes known as RT-qPCR, meaning reverse transcriptase real-time PCR. But we usually call it, for the most part, reverse transcriptase quantitative PCR, and it's quantitative in the sense that it can tell us how much virus or how much RNA was in our starting solution with respect to what was on off that swab that got extracted. So we'll talk about that a little bit here in a minute. But you need the instrumentation to actually read the amount of amplified product. And in this case, we eventually amplify DNA, and that's what is measured, and you need the appropriate machine and the technologists, the people who know how to use it, the microbiologists, the lab folks, in order to be able to do it. So the first step in this process is we got to do our sample collection. And again, it's that nasopharyngeal swab. Here's one of our recent graduates, Emma. She did her MPH here, Master of Public Health, with us. She actually did her capstone on Lyme disease with me and Dr. Brown. And she is now the County Public Health and Environment Director for Fremont County, Colorado. And that's her with her team. And they are inclusive of nurses and also potentially doctors, but most health departments have public health nurses that are able to actually take these kinds of samples. So they're doing drive-through sampling. Now, like the drive-through sampling, or if you were in the clinic with some symptoms and it warranted you actually getting a test done, what would happen to that swab? So that swab is going to then be subjected to an RNA extraction kit, where they're going to take part of that swab and we have to get off of that swab, and there's different steps to get things off the swab into solution. And then QIAGEN has a QIAMP RNA extraction kit, but there are other brands, Roche, and there's a whole bunch of other ones that have these kits where you can actually extract the RNA. So remember, coronavirus is an RNA virus. So we have to get the RNA off of that and then have it ready in solution for us then to be able to start our qPCR process. So when we do qPCR, and this is a very much a simplification, and sometimes you have different size tubes depending upon what you're using, but you have to have your initial RNA in the solution, whatever you've extracted from a patient, from an individual. We then have to add the enzyme reverse transcriptase, because the virus itself, needs that enzyme in order for us to then convert the RNA into DNA, because our qPCR, the reaction requires us to measure the amount of DNA. DNA is what we can amplify and make lots of copies of. That's exactly what the virus does. So the virus infects its host, and then it uses the host machinery to then create lots and lots of copies using DNA. So we have to have reverse transcriptase enzyme to then go along with our RNA. It has to have the right buffer in there, or the right pH, the right solution, so that we don't destroy the RNA. We have to have all the right oligosaccharides, to be able, and the primers that have to be in there that will allow us to make complementary DNA. And then the complementary DNA is then subjected to the usual process that we would do on most things like bacteria or DNA viruses. We then have to add this DNA polymerase, which, if you remember from biology, DNA poly helps us make lots of copies. So we make copies of the actual complementary DNA in the solution. It has to have the buffer. And in order for us to make the copies, we have to know, just like a zipper on like a jacket, you have to have the zipper lined up and before the actual zipper can run. So we have to have the primers for us to line up the DNA with a place where we know we can make copies, and then this thing can make the copies. And then when we make all the copies, it amplifies and makes lots and lots and lots of copies of DNA in our solution. This doesn't happen just automatically. Remember that machine? These machines are often associated with a thermocycler. Some have a thermocycler built in, some the thermocycler is attached. When I say thermocycler, in order for this to actually work, we have to raise and lower the temperature many times. So there's all these different steps for annealing and extension and all these things associated with amplifying the amount of DNA. And as we do this, raising and lowering the temperature 12 times, 14 times, 16 times, 18 times in this thing called the thermocycler, it keeps going on and on and on. And eventually, if there was a whole lot of actual starting RNA and then a whole lot of that complementary DNA produced from that, when it starts to get amplified at an exponential fashion, We will then get a fluorescence signal because of the kit that is used that the machine will realize. So a low cycle threshold hitting exponential copying at say 26 or 28 cycles means there was a lot of starting material there. If it takes to like 44, it might mean that there wasn't a whole lot, or we're amplifying something else that was kind of maybe similar. There was some sort of interference. So I don't know enough about the standard method and the actual calibration curves that are being used for this current kit, but we know that if you don't have any amplification, they're definitely clear, and there may be some point where these folks may be inconclusive. But if we amplify a lot of virus early on in the form of taking the RNA, the DNA, then we would know that that person had the virus in them at the time of the sampling. So there's a lot to that with respect to the amount of effort that has to go into doing the actual testing. So if you're going to be doing the testing, you're going to be a microbiologist, a person who's had genetics or advanced laboratory training. These are not easy skills to be able to get overnight. So it's important for us to understand that that's the current method. Easier tests, though, are being developed. If I were to pause this and I can pull up a little bit of information from Google Scholar right now, I think you'll be surprised. Okay, so now I've brought up another type of test just in the background here using Google Scholar, and I just typed in serologic test SARS-CoV-2, where we're actually looking at the immune response to ever having exposure. And since 2020, you can see that there's about 254 results for that. And if I do serologic tests for COVID-19, we see that there are 712 results. So this is something that provides us far more promise than just actually looking for virus, but rather looking for specific antibody responses to the actual virus. So that's where we're going to go, and most antibody-based tests are a lot easier to do than the actual coronavirus test using PCR. However, getting the actual antibody test ready to go and ready to run, that takes a lot more time in actually being able to pull that off. So we shall see. I'm going to stop this video here, and we're going to go back to what did we learn from our case series in Zhejiang province. So we will stop now.